The mitochondria. Everyone knows it, everyone loves it, and every cell needs it to survive. And even though the powerhouse of the cell is such a valuable organelle to the cell as a whole, one small injury to its structure can ultimately turn it against its cellular brethren. In the light of recent research, a relationship has been found between the mitochondria and the onset of cancer. The metabolic and bioenergetic functions of the mitochondria are vital to the health of a cell. So when mutations occur in oncogenes or proteins that are responsible for signal transduction pathways that interact with the mitochondria, or mutations occur within the mitochondrial DNA itself, disaster is certain to follow suit for the cell. Studies have shown that, in the tissues of tumors, there is an abnormally high rate of aerobic glycolysis taking place, which produces large amounts of glucose, which in excess can restrict the respiration of a cell, possibly causing it to turn cancerous in a process known as the Crabtree effect. Other data used to develop another theory, known as the Pasteur effect, has also indicated that an increase in oxygen levels can inhibit glycolysis. Whether either of these assumptions are totally true has yet to be fully confirmed by scientific research, but both ultimately support the idea that cancer is a metabolic disease. This fact is one of the major reasons as to why scientists suspect the mitochondria to play such a pivotal role in the onset of cancer. Cancer cells frequently are found to have mitochondria that are suffering from some form of structural abnormalities, which subsequently leads to defects within their functions. Whether it be alterations to the mitochondria's own DNA or proteins and lipid contents that are vital to the function of the organelle, a dysfunctional mitochondria can ultimately lead to a cascade of problems that, in turn, will more likely than not result in cancer. A key mitochondrial lipid, known as cardiolipin, is responsible for allowing proper cellular respiration to occur, and in cancerous cells, it is commonly found to be composed differently from abnormal contents than it is normally within a healthy cell. This information, along with the facts that cells display increased levels of anabolic reactions and below average conservation of pyruvate, further support the fact that a cancerous cell and its mitochondria are abnormally dependent upon the process of glycolysis. Cancer cells are unique in their metabolic processes. In many cases, even in aerobic favoring conditions, cancer cells will still undergo the process of glycolysis. In tumorous cancer cells, three key parameters indicative of mitochondrial function, which are cellular ATP production, mitochondrial potential, which is similar to the concept of water potential, but instead it involves hydrogen ions being harnessed to produce ATP, and cellular cardiolipin levels, are found to be 2 to 3 times higher when compared to non-cancerous cells. Mitochondrial mass also tends to be significantly higher in a cancer cell, and larger mass tends to be quite consistent with proliferation, which is the rapid growth or division of cells or certain parts of a cell. Another interesting fact to take into consideration is that when ATP production is inhibited in both a normal and cancerous cell, ATP amounts were decreased by only 30 to 40% in the cancerous cells, while normal cells saw a nearly 60% drop in ATP amounts. A cancer cell's ability to better withstand such an intrusion of ATP synthesis further supports the idea that they rely on glycolysis as a mechanism to produce their energy, implying that a dysfunctional mitochondria is a significant factor in the creation of a cancerous cell. Now, with all this information that has been acquired throughout the years of research into the mitochondria and its role in cancerous cell development, scientists have begun the daunting challenge of applying their knowledge to various forms of therapy in the hope of one day finding a cure for a disease that kills nearly 8 million people worldwide every year and sees another 14 million newly diagnosed individuals within the same time span. Data has led scientists to believe, with nearly full certainty, that glycolysis is the major metabolic process that produces energy and supplies anabolic growth within a cancer cell. Clinical tools have been produced with this information in mind, allowing for researchers to stumble across another key discovery: the role that mitochondria play in a process known as oncogenesis, which involves normal, healthy cells becoming transformed into cancer cells. Oncogenesis occurs after the activation of an oncogene, which results in cellular and genetic changes that lead to a cell dividing in an uncontrollable manner. The building blocks that are required for this mass division of cells and subsequent formation of a tumor come from the mitochondria of a cell. For this reason, the mitochondria has become a very promising target for researchers developing anti-cancer agents. If one has the ability to influence mitochondrial metabolism, then perhaps it may be able to influence and possibly halt the onset of cancer through therapy. Considering that the average mitochondria has only 37 genes in total, the relatively simple genetic makeup provides a manageable number of targets for researchers to experiment with. For reference, the human genome can contain anywhere from 20 to 30,000 genes, so the mitochondria is truly a simple and therefore attractive target for drug testing. With our current understanding of the relationship between mitochondria and cancer, researchers can say with general confidence that the mitochondria, in various ways, supports and contributes to the growth of a tumor in its four stages, which include initiation, growth, survival, and maintenance. On top of this, the mitochondria also plays a vital role in the pathways that are responsible for the growth of cancers, which include producing more mitochondria, mitochondrial fusion and division, cell death, which is also known as apoptosis, regulation of reactive oxygen species, and other communications that occur within a cell. All of these cellular operations, which are capable of being influenced by drugs, make good targets for researchers attempting to develop cancer eliminating therapies. The main reason as to why most victims of cancer are elderly is primarily due to the fact that, as humans and other organisms age, our mitochondrial DNA becomes damaged through various mutations that occur throughout our lives, leading to dysfunctional mitochondria and ultimately cancer. However, with all the renewed research occurring over the subject as of lately, scientists are beginning to get a fresh insight into all the mechanisms of metabolism that share a close bond with the mitochondria that frequently are found to be indicative of cancer. If we can translate this knowledge into effective treatments or drugs, then perhaps one day all humans can live in a world without having to fear the prospect of cancer.